Okay, hello everyone. I can see it's taking a little while to let everyone in, so just giving you a second to connect. A big warm welcome to our webinar today on what are advanced therapies, which is the first in a four-part series, which is giving you guys an introduction to advanced therapies and how they might be applicable in rare diseases. I'm Philippa and I'm Projects Manager at Beacon, and it's really wonderful to have you here for our webinar today. So firstly, for those of you who do need to use subtitles in Zoom, those are enabled for you today, and you can access these at the bottom of your Zoom screen. If you also don't want these on, you can also hide those in that bar too, and you can kind of control it from there based on your own preferences. A little bit of housekeeping. We're using Zoom webinar today, which many of you will be familiar with by now. If you have any technical difficulties and you want to communicate with Beacon staff or other participants, you can do that in the chat box on your screen, and you can type any questions into the Q&A box. Also, we'll be having a Q&A session at the end with our speakers, so that's a real opportunity for you to ask very specific questions. Make sure you put your questions in there, because if you put them in the chat, they might get lost. You can click on the thumbs up icon to upvote questions that you might want to see answered first, ones that you think are really important or that you also want to hear the answer to. So in case you're new to Beacon, we are a charity that's kind of pushing that no one should have their rare disease journey alone. This is because there's some key challenges that people living with rare diseases face, such as the diagnostic odyssey. On average, it takes a rare disease patient five doctors and 3 misdiagnoses before they receive their final misdiagnosis of their rare condition. Meanwhile, it's estimated that only 5% of rare diseases have licensed treatment currently. And in day-to-day life, there's a lot of challenges with things like care responsibilities and holding down paid employment. So to tackle these issues, we work directly with patient groups and patient group leaders. We do this through patient group training, which supports groups to form, grow, and professionalize, and empowers groups to provide emotional and practical support to their communities. Meanwhile, we also work within rare research to help put patient groups at the heart of research so that they can improve outcomes and access to treatments. We also facilitate connections across the rare disease community to strengthen the community on the whole. So just a little bit of background to why we decided to explore advanced therapies in this webinar series. It's something that you might have been hearing about more and more in the news, in media, seeing these amazing videos and pictures of people having access to advanced therapies and the life-changing impact that can have. But you might be thinking, what actually does it mean to produce an advanced therapy, and are they relevant to me and to my patient community? So this webinar series aims to provide a gateway of understanding that's really targeted towards you as patient groups, so you understand where you might fit into this process, help individual patient groups and representatives to better understand whether advanced therapy is actually viable for them and for their community as a route to treatment, and bring together a range of expertise and voices to provide that useful overview. So today's session is going to be looking at what advanced therapies are and what the term advanced therapies actually encompasses, what indications advanced therapies are being developed for, what the development landscape is looking like, and what we might be able to expect going forward with this relatively new route in drug development. What is the potential of advanced therapies in rare disease? And that's it. And we're joined today by 2 wonderful speakers. We've got Simon Waddington, who is Professor of Gene Therapy at UCL, and Jutta Griesenbach, who is a Professor of Molecular Medicine at Imperial College. So without further ado, I'm going to pass straight over to Simon, and he's going to give us his introduction to what advanced therapies are and their applications in rare diseases.
Thank you very much indeed. Let me just share my screen. Can you see my screen?
Yeah, that's perfect. Thanks, Simon.
Great. Let me just move people's pictures out of the way there. So thank you very much indeed for inviting me today. I'm going to talk about gene therapy, which I've been working in for about 25 years. In fact, actually, I think, Uta, I think I've known you for about 25 years, haven't I? I work in gene therapy for inherited genetic diseases, particularly those affecting children. And so I'll start off, first of all, by telling you how our cells work. And then I'm going to tell you about how viruses infect our cells. And there's a reason for this. And then I will tell you how we have exploited viruses for gene therapy. And then I'm going to move into what Hollywood makes of it all. And then I'll hand over to Uta, who will then describe some of the better detail about viral and non-viral gene therapy and ex vivo and in vivo gene therapy. So if I kick off here just by showing this video here. So this is a young girl called Amanda Cardoz. And here she's has type 1 spinal muscular atrophy. So at 18 months of age, she's got virtually no strength in her limbs because it's a disease of the motor neurons. And unfortunately, she passed away at 21 months of age, and she was characteristic of the prognosis for infants with type 1 spinal muscular atrophy. And it's just, these are the sort of diseases that me and my lab have been working to try to treat, and us generally as a field. So I just want to show an interesting paper that came out in 1966. So this is less than 15 years after the discovery that DNA was the material, the genetic material. It was also less than 15 years after Watson and Crick and Franklin discovered the DNA double helix. So this is 1966, and what Edward Tatum said, he said that in the area of metabolic disorders, the recognition of the genetic basis of many more disorders can be anticipated. Rapid, simple, and sensitive methods for the detection of carriers and for early diagnosis of affected individuals will be developed. So that was incredible that he was able to predict this in 1966. Well, then he went on to say that we can even be somewhat optimistic on the long-range possibility of therapy by isolation or design, synthesis and introduction of new genes into defective cells of particular organs. And then went on to say, finally, it can be anticipated that viruses will be effectively used for man's benefit in theoretical studies, in somatic cell genetics, and possibly in genetic therapy. And this was four years before even the first understanding of molecular biology. So even back in the mid-1960s, people were aware that this was a possibility. So I'm now going to move into a bit of a biology lesson. And I'm going to show you a simple cartoon here of our cells. So each cell in our body contains a nucleus, which contains DNA. The DNA makes up our genes, and so the DNA is packaged up into chromosomes. Each gene encodes a protein, and essentially the proteins are our nanomachines. They are the enzymes of our body. They also form the structure of the muscles. And what happens is that the DNA that's in the nucleus, it gets converted into a message called messenger RNA. And then that RNA shuttles out of the nucleus, and then it's decoded by these ribosomes and is converted into proteins. And these are the proteins here that you can see in pink. Well, what happens in a genetic disease is that you might have a mutation in one of your genes. And so when that gene gets shuttled out of the nucleus as RNA, it still contains that mutation. So therefore, the RNA can't be converted into a working protein. So that's what happens in a genetic disease. So I'm now just going to switch tack here, and I'm going to talk about viruses. So this is a virus that's just about to infect our cell. And the way that viruses infect our cells is that they attach themselves to one of various different proteins on the surface of the cell. So they've evolved over hundreds of millions of years. They've co-evolved to essentially to hijack our cells. What you'll get is you'll get a viral particle, and it will attach itself to the surface of the cell, and then it's able to transport itself and its genetic material into the nucleus of the cell. And so what you've now got is you've got the virus has injected its genetic material into the nucleus. And what then happens is that it hijacks the ability of the cell to make proteins. And so the cell starts to make viral proteins. And before long, what you have is that the cell has been hijacked to make billions and billions of virus particles. So essentially, the virus is there as a kind of parasite that exploits our own biological mechanisms to make more copies of itself. Now here I want to just show a comparison between a virus and a viral vector. So on the left, you've got a virus, and on the right, you've got a viral vector that we would use in gene therapy. The difference is that on the inside of a virus, the virus contains its own genetic material that allows it to infect the cells and allows it to replicate. But what we've done in gene therapy is that we were able to package just the therapeutic gene that we want to deliver, but on the outside, it is a virus, but just on the outside. And a comparison is that if you wear a leather coat, it doesn't mean that you're a cow. So here what you've got is that a gene therapy vector on the outside looks like a virus, but on the inside is a therapeutic gene we want to deliver. And what I want to show you now is here we've got a cell with a genetic mutation, so it can't make that little pink protein. And we've got our gene therapy vector that we're going to deliver. Initially, it's working just like a virus. So here, it attaches itself in just the same way to the outside of the cell. It makes its way into the nucleus, but this time it delivers working copies. It delivers functional genes. And so now, for the first time, the cell is able to make that protein for the first time. So this is the principle of viral gene therapy. Okay, so now what I want to talk about is Hollywood. And I'm going to show a few films which have featured gene therapy over the years. And not only is it just a bit of entertainment value, but there are some useful messages that I can talk about, and what Hollywood has tried to get us to believe, and also what the reality is. And if you want to guess what the films are, then please type them in the chat. If not, then maybe the panelists can guess too. So I'm going to show you the first film. See if you can guess what it is.
Last night, he killed a grizzly bear, the most powerful terrestrial predator on the planet. So you tell me right now, Kate, how any of this makes sense.
Are you familiar with CRISPR?
Yeah, genetic engineering.
It's more like genetic editing. I am talking about extremely specific results. Check this out. Look, sharks are indeterminate growers, right? They never stop. Isolate that gene and combine it with, say, the growth rate of a blue whale, the strength of a rhinoceros beetle, the speed of a cheetah.
And you get one confused gorilla that could take down a grizzly.
Rampage, yes, it is Rampage. So there we are, Rampage with Dwayne Johnson. So if we believe Hollywood, then The Rock knows what CRISPR is. But also if we believe Hollywood, then growth, strength, and speed are determined by a single gene, which of course we know is not true. Is that all of these attributes and many more are determined by many genes. And even some genetic diseases are quite complex. Different mutations in the same gene can cause different manifestations of genetic disease. So it's pretty complex. Okay, I'm now going to move on to the next film. Already guessed, Morbius. Very well done there. Yes, indeed, it is Morbius. Yeah. So if Hollywood is to be believed, then it takes 13 hours, 60 minutes to do molecular biology. And certainly as Uta and I know very well, it is really hard to isolate genes, to clone them into cells, and certainly to make vectors. It takes months or years to do this. It certainly doesn't take 13 hours. It's really hard. Also, the idea of just pulling out genes from one organism and expecting them to work in another is a little bit far-fetched too. Anyway, so let's move on to the next film. So indeed, it is Venom. Yes, that's right. And the take-home message here is that gene therapy actually spans many fields and technologies to treat many different conditions. So, you know, I and Uta work on gene therapy for inherited genetic diseases. But in this case, the film is featuring a gene therapy to treat cancer. So there are gene therapies to treat cancer, cardiovascular disease. There's genetic therapy for vaccination. So of course, you know, for the COVID vaccination, adenovirus vectors have been used, RNA vectors have been used. So there's a huge suite of different technologies to treat different types of conditions. So at the far end of the spectrum, you've got anti-cancer gene therapies. And here you're delivering genes that are designed to cause damage, to kill tumor cells, and also to cause an immune response. On the other end of the spectrum, you've got the sort of work that I'm involved in, which is treating inherited genetic diseases. And there you're trying to deliver genes as stealthily as possible. So therefore you don't cause any inflammation, you don't cause any immune response, and you're trying to put back a therapeutic gene. So we've got a massive suite of technologies which are all bundled together as gene therapy. And although in the field we all get together, we all get on very well, we're working on, in some cases, some quite different technologies. Okay, so next film. It's actually, I think it's Rise of the Planet for the Apes. Anyway, so according to the film here, gene therapies passed on to the offspring, because what I understand is that they deliver the gene therapy to the mother of Caesar, the main ape in the film, and this is inherited by Caesar. So one crucial point to make is that gene therapy only treats somatic cells of the body. It is prohibited, a worldwide moratorium on germline gene therapy. And if you remember, there was the Chinese scientist that did a genetic therapy for those twins to make them resistant to HIV. And there's a huge furor of this because there is an international ban on this. So gene therapy is designed only to treat your body cells, but not to be transmitted to your offspring. If we believe Hollywood, then you can just aerosolize your gene therapy and it will transduce your brain and every cell in your body. I'm sure as Uta will be very happy to explain, working on gene therapy for cystic fibrosis, it is not that easy to deliver genes as an aerosol, because our bodies, certainly for viral vectors, our bodies have great antiviral defense mechanisms. And since the vectors on the outside look like a foreign particle or even a virus, our bodies are designed to eliminate them. So trying to get them in is really hard. And this is why it's taken so long for even some of the easier gene therapies, because it has taken so long for us to get the technologies to a level of efficiency that we can deliver effectively to certain cells. If Hollywood is to be believed, then it will make you super intelligent, but only if you're an ape. Okay, so next film, and these are getting older still. Yes, I Am Legend. Very good. Yes, I Am Legend. Yeah. So if we believe Hollywood, then gene therapy vectors often turn into super virulent viruses, converting everyone in the world into flesh-eating zombies. So the first point here is that in gene therapy, when we have manipulated viruses to turn them into gene therapy vectors, what we're doing is stripping out all of the viral genes, some or all of the viral genes. So inevitably what we're doing is we're breaking the virus, which means the virus doesn't work anymore. So it's not going to get worse, it's going to get broken. And particularly for gene therapy for inherited genetic disease, you want as little of the original viral genetic material in there as possible. So one of the vectors which has proved to be most useful is a thing called AAV, which is adeno-associated virus vector. And here it has got about 5% of the original viral DNA. It doesn't make any of the viral genes at all. So there's no virus left there. So that's a really important point. The other point actually is one that I made earlier, which is that here they're talking about an oncolytic virotherapy. So here they're trying to destroy tumors, which again is at the opposite end of the spectrum to that which Uta and I work on. Okay, so that was I Am Legend. Right, next film. Yes, very good. Not only Bond, but Die Another Day. That is correct. Yes, very good. So it is Die Another Day, and apparently, according to Hollywood, ex vivo gene therapy is where bone marrow is taken from orphan strays and runaways to be injected into the patient and is known as DNA replacement therapy. So I know I forgot to mention yes, I forgot that I was going to talk a little bit about ex vivo and in vivo gene therapy here. So first of all, obviously, is that in doing gene therapy, we don't take bone marrow from orphans, strays, and runaways. So the point here of this that I want to make with this film is that there is in vivo gene therapy, where you're taking a gene therapy vector and you're injecting it directly into a person. And then there's ex vivo gene therapy, where you are taking cells out, you are treating them in a dish with a gene therapy vector, and then putting them back in again. And a good example of this is X-linked severe combined immune deficiency. So these are infants with a mutation in their immune system. So stem cells, their immune stem cells are taken out, the working gene is inserted into the stem cells, and the stem cells are reintroduced into that infant. And they then essentially grow a working immune system. And the beauty of this is that essentially is that they are their own bone marrow donor. So therefore, it is much more likely that the graft will take compared with the previous standard, which was a bone marrow donation. Another important point is that the idea of calling it DNA replacement therapy. So with gene therapy, mainly what you're doing is you're putting in extra working copies of the gene. So you're not editing the genes, you're not taking them out and putting in new ones, you're just putting in more copies. Now, I think Uta might talk a little bit about gene editing, which is kind of a little bit more finessed, but the bulk of gene therapy is just putting in extra copies. And it's rather like a kidney transplant. So recipients of kidney transplants, they don't tend to have their kidneys, the diseased kidneys taken out. They just get an extra kidney put in. So that's basically the principle of most of gene therapy at the moment. Okay, I think we've got one more film. It's The Bourne Legacy. So that really wins a gold star, because Rachel Weisz's character, she says, getting the gene where you want it is the nightmare. And that really is still the case in gene therapy. It's still difficult to be able to target to exactly the cells that are affected. We're getting better at it over time, but really it's still work in progress as we improve with our ability to get the DNA or the correction to the right cells. The next point is we're still trying to really understand how the gene therapies work. I mean, we know quite a lot, but there are still areas where we don't understand things. So again, it is still it's a whole field which is still learning and still developing. I've seen a question there, so I'll take this now. So the question is if putting in good DNA, when is the target to remove the bad DNA? So this is kind of refers to what I was saying about the DNA replacement. So what generally happens is that you've got a broken gene, the gene isn't doing anything, it isn't making anything. So what you're then doing is you're putting in a working copy of the gene. You're not taking out the one that's not doing anything because you don't need to. You're just putting in a working copy. These tend to be the easier diseases to treat, like haemophilia, where haemophiliacs can't make the clotting factor. So therefore, you're putting in working copies. But what's happening is that now, as the technology is improving, we're starting to move towards harder and more complex diseases, where we might want to try to shut down a bad gene as well as introducing a good gene. Okay, I'm just going to continue with the talk just so I don't lose my thread. So here I just want to make a brief comparison between the different types of treatment for example, inherited genetic diseases. So you have drug treatments. So for example, for alkaptonuria, you might have nitisinone. And nitisinone has to be given very regularly, because drugs don't last very long. They might only last for a few hours or a few days. Then you have things called biologics, and these are biologically produced compounds such as antibodies or interleukins. And so for haemophiliacs, people with haemophilia, conventionally they've had to inject themselves with factor IX protein or factor VIII protein. Factor IX only has a half-life of about 18 hours, so it doesn't last very long. It lasts for days. It certainly doesn't last for weeks. We're now starting to see RNA therapeutics. And if you remember the graphic that I showed you right at the start, RNA, our cells make protein from RNA. So RNA has been found that in RNA therapeutics, it's been found to last a bit longer. It seems to last longer than some biologics. In some cases, it's lasting days or weeks, or its effect might even last for a couple of months, but it's not permanent. And then we have gene therapy vectors. So for some diseases, these appear to be lasting for years and years. So people with haemophilia B, they've received a single injection of a gene therapy vector, and they are now making factor IX, the protein that's missing in haemophilia B, and they are making it 5 years later. So those last a very long time. And then we have stem cell therapies. In those cases, and again, I'm making it much simpler than it really is, stem cells, in theory, should last for a lifetime. And so there have been attempts at doing stem cell therapies, for example, for Parkinson's disease, where you're trying to put back into the brain the dopaminergic cells that have been lost in Parkinson's disease. So roughly, this is what you're looking at here, is that drugs don't last for a very long time, long at all. Biologics a bit longer, RNA therapeutics a bit longer, the viral vectors longer still, and possibly 50-50 between them and stem cells for lasting longest of all. But different diseases are treated with different modalities. I'm now going to just loop all the way back to type 1 spinal muscular atrophy. So as I mentioned, it's an inherited neuromuscular disease. Motor neurons die. Infants will never sit unassisted or walk, and they will rarely reach their second birthday. So when natural history studies have been done on infants with this disease, this is how it looks. So therefore, on the left-hand side, you see a graph of the motor ability of infants with type 1 spinal muscular atrophy. And unfortunately, untreated, there are no treatments, there's only symptomatic management. And their motor score will decline over time, and many infants with type 1 will not live beyond 2 years of age. And generally those do survive will have a gastrostomy or a tracheostomy, or they will have 16 hours of non-invasive ventilation a day. So it's a pretty dismal disease. And then around 2010, there was a paper which showed that a vector injected intravenously into a mouse model of type 1 spinal muscular atrophy would extend the lifespan. So these mice, untreated, would die at around 14 days of age, but with the gene therapy, they would live for up to a year. So it took more than half a decade to take this to clinical trial. They made enough vector to treat 15 infants. So 3 received the lower dose, and then the remaining 12 received the higher dose. These were all infants with type 1 spinal muscular atrophy, and they were treated on average at about 3 months of age. So if you just bear in mind what you can see on the left-hand side there of the motor score, and now what I'm going to show you is the graph of the motor score of the infants that received the gene therapy. And you can see here that all but one of them improved over time. They all exceeded the previous best ever motor score for an infant with type 1 spinal muscular atrophy. And if you look at the very top left-hand quadrant of that graph, you can see 2 infants there that exceeded a motor score of 60, which means that they were indistinguishable from infants who do not have spinal muscular atrophy type 1.
Now, 3-year-old Evelyn challenges Dr. Mendel to a dance-off at her annual follow-up appointment.
She comes back after 3 years and she runs up to me and hugs me and says, Dr. Mandel, I love you.
I forget that she has SMA. This is a healthy girl and she does everything that a normal 3-year-old child would do.
So that's Evelyn, and she was one of the infants on that trial that I showed you there. So now in the EU, these are the medicines that have been approved by the European Medicines Agency. The most recent approvals were for Upstaza, Roctavian, and Hemgenix for a disease called aromatic acid decarboxylase deficiency, haemophilia A and haemophilia B. And Uta may go into this more, but some of these diseases were, in theory, some of the easier diseases to treat, which is why they have now reached market approval. So it takes years to come from the point of doing the mouse studies to the point where you can do a human clinical trial, and then it takes several more years from the point of doing the clinical trial to the point of market approval, if you can get it at all. So it is a really long journey. From the early experiments. So as I said earlier, we're trying to treat in gene therapy a huge range of different diseases. We're using genetic therapies as vaccinations. We're trying to treat solid cancers and blood cancers and cardiovascular disease and inherited genetic disease. And to do this, we're using a whole range of different technologies. We're using viruses, non-viral technologies, stem cells. So I want to kind of leave it at that point there, really, to say that the gene therapy is complex and wide-ranging, and isn't just a single technology, and it's specific technologies for specific indications. So I just want to wrap up there. Hope that I've explained a little bit about how our cells work, how viruses infect us, how we've used viruses for gene therapy, and also a little light entertainment of what Hollywood makes of it all. And I think now I'd like to thank you for your attention. And do I now hand over to Uta?
Thank you so much, Simon. And yes, Uta is ready and raring to go. Consider me entertained, and hopefully everybody else is entertained as well. A really brilliant introduction. And as we said earlier, Simon will be around at the end to answer any of your questions. And now I'd love to hand over to Uta, who's going to be presenting a little bit more about what Simon talked about, and also what the pipeline of development is looking for gene therapies at the moment. So Uta, over to you.
Thank you, Phil. Can you see my slides all right?
Yeah, it's perfect.
Great. Good morning, everybody. So I'm a professor of molecular medicine at Imperial, and similar to Simon, have worked in the space of gene therapy for over 25 years and have really over, almost 3 decades seen the field grow fantastically, and I hope you'll agree with me when we come to the end of my talk. I'm also a director of the Cell and Gene Therapy Catapult, and the Catapult is a taxpayer-funded company whose remit it is to grow gene and cell therapies in the UK. So a few years back, the UK government has made a strategic decision to really grow the industry of gene and cell therapy, and the field is very much benefiting from this investment of taxpayers' money into the space. Now, I can, of course, only give you a brief overview on opportunities and challenges that we are facing in the field of gene therapy. And it's always difficult to speak after Simon, because he's such a tough act to follow. And I certainly don't have any movie clips, for which I do apologize now. Some of my slides are also what you might perceive as a little bit UK-centric, but I think, the things that I'm saying are probably extrapolatable. The reason why I talk sometimes about the UK is because I know the infrastructure and the space in the UK the best. So what are advanced therapeutics and gene therapy? So gene therapy is what we call an advanced therapeutic, and just wanted to sort of define this because sometimes the nomenclature gets a little bit confused. So advanced therapeutics or advanced therapeutic medicinal products it's the same thing. And into this category falls gene therapy, but also cell therapy, so gene modified cells, as well as tissue engineered products. Just briefly going over what Simon already said. I just need to move the bars here a little bit. I just wanted to discriminate the ex vivo and in vivo applications. So when we're talking about ex vivo gene therapy, it means that we're taking cells from the body and we are modifying these cells in a Petri dish. And once they're modified, they are retransplanted into the patients. And examples here are anti-cancer therapies that are based on gene therapy, as well as gene therapy for immunodeficiencies. And I'll go into this a little bit more later. When we talk about in vivo gene therapy, we mean that the gene transfer agent, our viral vector, for example, is directly injected into the patients, either intravenously into the spinal fluid. Sometimes we do muscular injections. And in my research field, which is gene therapy for lung diseases, we deliver the gene therapy agents via inhalation. So the examples here are the things that Simon mentioned: spinal muscular atrophy, hemophilia, and cystic fibrosis. Now, when we talk about gene transfer agents, it's just important to understand what we mean. Gene transfer agents fall broadly into two different categories. One category is based on using viruses, viral vectors, and we have a variety of viral vectors that we are using. They all have fancy and complicated names. They all have slightly different properties. When we talk about non-viral vectors, these are generally much simpler complexes or configurations where we take the gene, which is on a piece of DNA, and we mix this gene with, for example, a lipid. So just a simple fat particle, and this fat particle helps the DNA to enter cells. Now, viral, non-viral vectors have a lot of disadvantages and advantages that I don't have time to go into, but I'm happy to answer questions about later. Now, the other thing I just wanted to highlight, and Simon alluded to this, when we talk about a viral gene therapy vector, it's not a virus anymore. And this is really important because a lot of the viral vectors that are currently used are based on a lentivirus. And some of you might know that HIV viruses are the most, or that many of the lentiviral vectors we are using are based on HIV viruses. So if you are affected by a disease, or if you are the father or mother of a child with a rare disease, and if I, as a scientist or clinician, tell you I'm going to treat your child with an HIV-based virus, you might be worried and you might be very hesitating. So it's really important to understand that all these nasty blue genes here, which make an HIV virus cause AIDS, for example, are removed in our viral vectors, and they are replaced by the therapeutic gene of interest. So, for example, in work that we do with the cystic fibrosis gene. The other big difference is that these viral vectors, once they are inside the patient, they don't replicate anymore. They just enter the cells, they sit there quietly, and they do not multiply and amplify like the original wild type viruses did. So in general, these viral vectors are considered to be safe. Now, since the early 90s, gene therapy certainly had a slow start, and it took us years to establish proof of concept that gene therapy works. And this was because we were on a very steep learning curve. We didn't know which of the viral vectors or the non-viral formulations we should be using for specific genes. But now we are in a much better position to understand, what is the best vector for an acute disease or for a chronic, lifelong genetic disease. We can choose if we want a gene therapy agents that give us very long duration of gene expression, which is important for chronic, lifelong diseases such as cystic fibrosis. we need to adapt the gene transfer agents depending on whether we want to put genes into dividing cells or non-dividing cells in the body. We need to be thinking about ex vivo and in vivo gene therapy, and the size of the gene is also really important because not all of the viral vectors can carry large genes. So I think what I'm trying to say badly here is that over the last 25, 30 years, we have learned an awful lot of how to best map our gene therapy tools to the actual disease indications that we're trying to treat. Just wanted to briefly talk about the difference between an integrating versus a non-integrating vector. So this schematic here is a chromosome, which we carry in the nuclei of our cells. Many gene transfer agents that we're using just sit next to our endogenous chromosomes. They don't integrate into our native DNA. However, we're also using integrating vectors, which do integrate into the chromosome and bury themselves away. Sometimes this integration has advantages because it means that the gene therapy product is produced for longer. So integrating versus non-integrating vectors is always a decision we have to make when developing a new product. I think in the interest of time, I'm just going to skip over this. Now, a little bit about the history and the problems encountered. So in the 1990s, the concept of gene therapy, became very, very popular, and over 10 years up to the early 2000s or so, there was a big hype about gene therapy, and everybody thought gene therapy will cure all diseases known to man very quickly. However, then there was a loss of faith in gene therapy in the next 10 years, and people felt that we really don't want to give gene therapy to anybody. We don't believe in gene therapy, and that was triggered by lack of efficacy in many, many studies, but also sadly by the deaths of some trial participants and side effects that were encountered. However, I think now we are in a position where we have much more realistic expectations what gene therapy can do. We have learned what our gene therapy vectors are doing and what they can't do. There have been lots of innovations, and as Simon said earlier, gene therapies are now becoming licensed medicines. So now just a few sort of statistical facts. This is a slide from the American Society for Gene Therapy, which produces reports every quarter. And to date, we have approximately 25 gene therapies that are approved worldwide. This doesn't mean that they're all approved in the EU and in the UK, but this is sort of a worldwide picture. In this report, which is widely accessible, it also lists all the different indications. I'm going to talk a little bit about a couple of those. One of them is Spremvelis, which is a gene therapy that was developed for children which have immunodeficiency, so children that can't fight infection. I'm going to briefly talk about two gene therapies that were developed for cancer gene therapies. Briefly going to mention a gene therapy that was developed for blindness, and then we'll talk a little bit about gene therapies for hemophilia as well. Now, this slide shows that over the last 5 or 6 years, the number of commercial companies interested in the space of gene therapy has very significantly grown, which is fantastic. The fact that pharma industry and biotech companies are interested in gene therapy is a real booster for the field. What you can see here in blue is that the number of academics and non-profit organizations working in this space has been pretty stable, but has certainly not grown. And sometimes I think being an academic based on the university, this is a little bit of a concern for me, because a lot of the initial innovation and the initial ideas are generated in industry and then sold to companies to be further developed. So one thing that I often say and like to point out to funders is that we need to do whatever we can to keep the pipeline of innovations coming out of universities alive. And I'm sure Simon will speak to this later as well. This is really, really important. The standard route from how materials are developed in industry is that we establish early proof of concept studies within the universities. We might do an early phase clinical trial, but then we either have to sell our products to an industrial partner, or we have to generate a spin-out company to further develop the product. So there's a whole complicated path and procedure to this. This slide here shows the number and the sort of distribution of UK gene therapy trials into different therapeutic areas. And you can see that the largest proportion here is oncology, which is really not surprising. Ophthalmology, so eye diseases and blood disorders also form very big chunks of our activities. My specialty, the respiratory gene therapy, only forms a small proportion here. And this is, I think, due to the fact that over years, it has been demonstrated that gene therapy to the lung is actually very difficult, because the lung is an organ that can fight gene transfer agents very well and isn't really susceptible, not easily susceptible to taking up gene transfer agents. So a lot of industry and to a degree academics over time lost interest in gene therapies for the lung and moved into slightly easier targets. But I think we are now in a position where lung gene therapy has also shown to be workable and has shown a degree of success. I think I'm just going to skip over this. Now, an important aspect of development for gene therapy is the growth of the industry. And this slide here just shows you a couple of sort of statistics where if we look at investments into advanced therapies, including gene therapy over the last few years, you can see that the investments are very, very significant. Manufacturing of gene therapy products is always a big issue. And you can see that in the UK, the manufacturing space over the last few years has almost tripled. The number of jobs in gene therapy related research is going up and up, and the number of clinical trials. So what I'm trying to say with this slide is that gene therapy is a very active and fast growing field. One thing we are often asked is, why does it take up to 15 years, sometimes longer, to translate an idea from the bench into what's ultimately a licensed treatment? And I don't want to come up with too many excuses for why it takes so long, but just illustrate the number of steps that are required. So within our laboratories, we might have an idea, we might have a hypothesis, and we then have to do preclinical studies in cell culture models or in animal models to establish proof of concept that our hypothesis is correct. And more often than not, we show that our hypothesis was incorrect. So we have to go back to a blank page and generate new hypotheses. We then also have to prove that whatever approach we are taking in our preclinical research is safe. So we have to do toxicology studies. The next thing we then have to worry about is the manufacturing of our gene therapy products. And there's a whole, I can talk for hours about challenges related to manufacturing, but it's not a trivial process to scale up our gene therapy agents to the quantities that we need. And there are a lot of aspects that we have to consider. We then need to think about delivery, and the delivery of our gene transfer agents probably pretty straightforward for intravenous and intramuscular injections. But in my field, where we need to target the lung, we have to use nebulizers, and we have to worry about whether our gene transfer agents are stable in the nebulizers, and whether, you know, the droplet size that we're generating is right to get to whatever airway sizes we want to go to. So the whole years of development just to get the delivery right. And then we have the different phases of clinical trials. And on this slide, actually on the next slide, I'm going to talk about clinical trials. Here, I just also wanted to tell you that before we can do a clinical trial in patients, there's a huge amount of paperwork. Every country has their own regulatory body. In the US, it's the FDA. Here in the UK, it's the MHRA. In Europe, it's the EMA. And if you want to do a clinical trial in these 3 countries, you have to speak to each of these regulators and get individual approval for your clinical trial. And the remit of the regulators is to review how the gene therapy product or the advanced therapies have been made. They have to review all the preclinical efficacy and toxicity data, and they have to review your clinical trial design to agree that whatever you're proposing to do in the patients is actually the right thing to do. And the key questions that the regulatory bodies are asking us is whether we have done everything possible to minimize the risks to the study volunteers. They'll look about the trial design, and they also look at whether the investigators who are doing the studies have the relevant expertise. So before we get to testing anything in patients, there are a lot of mechanisms to make sure that our strategies are safe and have enough evidence of proof of concept in animal models to allow us to go forward. And then there are many phases of clinical trials. So traditionally, a phase 1 clinical trial is a dose escalation study. And if you just use a biologic or a small molecule drug, for example, paracetamol, you would do a phase 1 clinical trial in healthy volunteers and generally in adults. Now, that's not the case for gene therapy trials. Gene therapy trials are almost never done in healthy volunteers. They're always done in the affected disease population. And if you are looking at a disease where, for example, children would die if they're not treated, you can do early phase clinical trials in children if you don't have any other options. Usually these are dose escalation studies, and they're often linked to assessing safety. And the design is often a design where you treat 3 patients with a low dose. If these 3 patients respond well and don't show any toxic side effects, you would then go to the next cohort, which gets a higher dose, treat another 3 patients, and then do a final high dose cohort, which would be your maximum feasible dose. Often these trials are double-blinded and placebo-controlled, which means the patients don't necessarily know if they have been treated with just a buffer or with the active drug. But sometimes these trials are also open-labeled, which means that both the patients and the doctor knows if the patient was treated with a drug or with a placebo. Once you have successfully completed your early phase 1, 2A clinical trials, you move into something called a phase 2B study. And these phase 2B studies are often still single dose studies. And in these studies, you would now use your maximum feasible dose. So in your phase 1, 2A studies, you've determined which is the best and the highest dose you can give that is safe. You then use this dose in the phase 2B studies. Phase 2B studies are usually a larger number of patients, and you're starting to look not just at safety, but also at clinically relevant endpoints. So in my case, for the lung, that's changes in lung function. And the phase 2B studies are more often than not double-blinded, placebo-controlled. If you're lucky enough that your hypothesis holds up in a phase 2B study, you then move into a phase 3 study. And this is now large number of patients to make sure your treatment really works in a variety of settings and scenarios. If your phase 3 studies are successful, you can license your drug. And You then have to do some post-licensing follow-up as part of phase 4 studies. So I hope this gives you a bit of a feeling for why it takes 10 to 15 to 20 years to get from an inception and an idea and a hypothesis to a licensable and licensed drug. A couple of examples that I want to give you, and I just need to quickly check the time to make sure we're not running out of time. Example here are CAR T cells that you might have heard of. And this is a gene therapy that was developed for cancers. And what you need to know is that some tumors make very specific proteins, which are called antigens. And these specific antigens are only or largely only present on the tumor cells. So what we're doing with these therapies is we're taking immune cells and we arm these immune cells to specifically recognize the tumor antigens, and then these immune cells or T cells kill the tumor antigens. And the way this works is that we take cells from the patients, we isolate these cells, we then treat them with a gene therapy vector that arms the cells, and they're now expressing specific proteins on the surface that attack the tumor and kill the tumor. These cells are then amplified in a petri dish and retransplanted into the cancer patients. And over the last few years, several of these CAR T-cell licensed products have been developed. One that you might have heard about is Kymriah or Yescarta. And these are very successfully used for lymphomas at the moment. I think future challenges for CAR T cells will be to use these CAR T cells for solid tumors. There are a lot of people trying to work up the technology, but at the moment it's proving rather difficult. The other thing that's currently happening is that these therapies are based on taking patients' own cells, so autologous cells from the patients, and modifying them. And this needs to be repeated on a patient-by-patient basis. And that's not a great way to develop a drug. You know, it would be much better if you could use donor cells, which people could donate, which could be modified, which could be frozen, and then used in a number of patients. And that would be much better approach from a drug development point of view, because it would be much cheaper. And that's also an area that a lot of people are working on. Just an example on a gene therapy for blindness, a disease called Leber's congenital amaurosis. The drug here that has been developed fairly recently is called Luxturna, and it's a viral vector, viral gene transfer vector that's injected into the retina. And this has certainly not been a cure for patients, but has very, very significantly improved the vision in affected people. Another example to briefly mention is hemophilia, and Simon talked about this as well. You can see that this drug Hemgenix was licensed just earlier this year in 2023. What I would like to point out here is the sort of headlines that this drug generated. So when it was licensed, the headlines were that it's the world's most expensive drug and costs approximately $3 million to treat a patient, and it's a very scary number. But you need to put this number into context, and the context really is provided here. So the company that generated this drug is CSL Behring, and they said that even though the drug is very expensive and costs $3.5 million, that over the lifetime of the patient, the drug saves the US health system roughly $5 million per person. And this is due to the fact that if you don't treat a hemophilia B patient with gene therapy, you have to treat this patient with a protein, with a blood clotting factor. And if you do that with a non-gene therapy-based treatment, you're spending roughly $800,000 per patient per year. So you can very quickly work out that over the lifetime of the patient, 3.5 million for gene therapy is a lot cheaper than the protein-based therapies, which are also not as good. So that's all fine. And I come a little bit back to these challenges later on. Another example that I just wanted to briefly mention are the immunodeficiencies. So ADA SCID is a disease where babies are born without an immune system, and unless they're kept in a very sterile environment, they very quickly succumb to infections and inflammations. It's an ultra-rare disease, which approximately 1 in 500,000 babies born having the disease. And the gene therapy that was developed for this disease is an ex vivo gene therapy, where bone marrow cells were taken from the baby. They were modified using gene therapy vectors in Petri dishes and then retransplanted into the patient. And this drug was licensed in 2017. However, there's a little bit of a story to this, which I'm going to try and tell you now. And it's a story that's a little bit concerning for people involved in developing gene therapy for rare and ultra-rare diseases. So the initial hypothesis was and developed and proof of concept established at the San Raffaele Institute in Italy in the early 2000s. Then GlaxoSmithKline, which is a big pharma company, became interested and started to collaborate with the San Raffaele Telethon Institute and the San Raffaele Hospital in 2010. And this was a very successful collaboration. They also brought in a company which is called MolMed to sort out the manufacturing processes. So this was a really, really good collaboration of fantastic groups to move this forward. As I said earlier, this condition is very rare and affects only about 14 people per year in Europe and costs roughly 600,000 euros per treatment, and obtained market approval in between 2015 and 2017. So in the same year as GSK got market approval, they also announced that they were looking to sell off Strimvelis, which was very surprising because they had only just obtained market approval. But nevertheless, whatever reasons, GSK decided not to move forward with this drug. Thankfully, in 2018, a UCL spinout, which is called Orchard Therapeutics, bought Strimvelis, which ensured that the drug could be supplied to patients to patients who needed the drug. However, in 2022, in the spring 2022, Orchard suddenly announced that they are cutting their investments into gene therapy. There were financial issues, and that they would discontinue investment into Strimvelis. So now you have a situation where a very successful drug that really cures babies and children who would otherwise not live was not available anymore. And thankfully, a non-for-profit organization, which is the Fondazione Telethon in Italy, has taken responsibility for making Strimvelis available via links to Italy. But this is really quite a sad situation where a non-for-profit organization now has to step in to provide a drug for an ultra-rare disease. And sadly, that's what we're seeing more and more, that the big pharma companies are getting less and less interested in ultra-rare and rare diseases, in part related to, I think, the profit margins being not as high as the companies would want them to be. Now, costs are an issue, and I mentioned the 3.5 million for the hemophilia drug. And what we need are innovations in pricing models, because I think what insurances and in the UK, the NHS is suffering from is these very, very large upfront payments that are required. And there's a lot of debate, a lot of discussion on how we can manage the payment mechanisms to make treatments that are initially very expensive, but in the long term, over the lifetime of a patient, save money to the healthcare providers, how this can be adopted and managed. A lot of other innovations is happening in the field of manufacturing because the high costs are to a large degree due to the very high manufacturing costs, and a lot of people with very big brains are trying to work up manufacturing strategies that are scalable and cheaper. And I think over the next 5 to 10 years, a lot of innovations will be made in this space. The other challenges we are facing is, what are the risks of environmental exposure to gene therapy agents? And by this I mean, if you imagine a situation where you're treating a patient with cystic fibrosis, for example, through an aerosol, and then a few hours after gene therapy, the patient goes home on the bus and sneezes, how much viral vector is released into the environment, and what is the environmental risk? And these are all the things we have to consider when we are developing gene therapies, and we have to prove to the regulatory bodies that the risks are minimal. And if the risks of environmental exposure are not minimal, it means that maybe we have to keep the patients in hospital after gene therapy for a few days, maybe a week or so, until they're not releasing vector anymore when they're sneezing, which would reduce the environmental risk. Another challenge in the field that you might be aware of is patient follow-up. So for certain gene therapy products, we are required to follow patients for 15 years. And this is clearly a big, big burden, not just for the clinicians who have provided the gene therapy, but also for the patients. And to reduce the burden wherever possible, these annual follow-ups for 15 years are linked to standard clinical care that the patients receive. But these requirements for long-term follow-up is certainly, again, an area that is very much exercising the field, and where we're talking about mechanisms to make this long-term follow-up feasible for patients, but also for the clinicians and the sponsors. So before finishing, I've just got a couple of more slides that I wanted to briefly talk about. One other challenge to gene therapy are also the side effects, and side effects occur, any drug has side effects if used in high quantities, but with respect to integrating gene therapy vectors, there's a particular concern that we have, because a vector might integrate into the chromosome and could potentially cause damage to the chromosome or alter genes that are on the chromosome where the vector integrates, and this could potentially cause cancer. And I'm just going to tell you another story, and I'll let you judge if this is a success story or a failure. And it's a story that occurred, or a situation that occurred probably close to 15 years or so ago now. Was again an ex vivo gene therapy where children with an immunodeficiency were treated with a first generation vector. And this is important here, first generation vector. So ex vivo gene therapy, again, the bone marrow was taken, was modified, and retransplanted into the patients. And the story was that these numbers might be slightly outdated, but if you just bear with me, there were 20 children with SCID, with a severe combined immunodeficiency, were treated, and these children would normally not have lived very long because of the lack of immune system. And in about 85% of children, so in 17 out of 20, the disease was completely corrected. and the children lived a normal life, could go to school and to kindergarten and so on. However, there were some side effects. 5 out of the 20 children, so about 25%, developed a leukemia because the vector had integrated into the chromosome and caused a gene to be upregulated and caused cells to divide in an uncontrolled manner, which is basically leukemia. So 4 out of 5 of these children were very successfully treated using standard anti-leukemia drugs and are well. But sadly, 1 out of the 5 children died due to side effects of the leukemia. And when this happened, it was a big, big blow to the field of gene therapy, because all of a sudden, everybody got very scared of gene therapy, and a lot of clinical trials at the time were stopped. However, once the field started to think a little bit more about this, it became clear that the mortality rate of this gene therapy was 5%. So 1 in 20 children died. Now, the mortality of these children, if untreated, is 100%, right? So the 5% mortality due to gene therapy is all of a sudden a lot lower than the 100% if untreated. And an alternative option for treating these children with immunodeficiencies is to use bone marrow transplants. If you have a matched bone marrow transplant, your mortality rates are pretty low, but many children don't have matched transplants, and if they're treated with a mismatched transplant, the mortality rate is 50%. So if you look at these numbers, all of a sudden, you probably appreciate that, yes, there is a risk of side effects, but the risk is a lot lower than if patients were to remain untreated. So it all comes back to balancing the risk and the clinical benefit. And this clinical risk benefit analysis is very, very disease specific and very, very much dependent on the situation and the severity of your disease indication. Now, another challenge in the field, and I think I'm almost finished here, is there's currently not enough trained staff within the NHS to help with the development of gene therapies as part of clinical trials, but also with the delivery of a gene therapy-based medicines once they are licensed. And there's a lot of initiatives in the UK trying to upskill NHS staff and allied healthcare professionals to help with this space. And then patient and public involvement is also a big area within the gene therapy community. And here, it's really important to involve patients and the public into discussions about gene therapy products and clinical trials really early on. So I think with this I conclude, and just based on challenges for the future, I think the cost of gene therapy-based medicines has to come down for the field to grow further. I think we are more and more transitioning now from the rare and ultra-rare diseases to more common diseases, and I think this is in the space of more common diseases is where a lot of innovations will be made in the next 5 to 10 years. We need innovation in manufacturing. We need innovation in making our gene transfer agents even safer. And, we need to worry about the workforce who is going to deliver the gene-based medicines once they're licensed. Just in terms of information, if you are interested in gene therapy and want to get more information, there's a really nice European website, which is called EuroGCT, which has a lot of information for patients and the public related to gene therapy topics. And certainly the societies, the British Society for Gene Therapy, the European Society and the American Society on their websites also have a lot of information on gene therapy for patients and public. And I think with this, I will stop to make sure we have a little bit of time for questions. Thank you very much for your attention.
Thank you so much, Jutta. Such an in-depth and well-varied presentation with all those challenges and, you know, what we can expect from the future as well. I'm just going to ask the speakers to turn your cameras back on. Simon, if you can do that, we've had a question come in. And also I've been thinking of a few questions myself that you've kind of stirred in my brain. But firstly, somebody has asked, I've heard that if a patient has gene therapy, they are reclassed as a modified body. And according to Department of Pensions, the patient then cured no longer eligible for benefits. Now, this is obviously looking at the actual impact of their life. But is that something that you've heard, or are they still kind of classed in the same categories before?
I'll pass on to Simon in a minute, but I've never heard that a patient who had a gene therapy-based treatment is not eligible for benefits. I doubt that that is true. Simon.
No, I've not heard that either. I mean, what sort of benefits and what was the possible reason for this?
You'd really hope that wasn't true. So hopefully,
that sounds really weird.
Yeah.
I think what is true is that once you had gene therapy with a particular gene, say if you have cystic fibrosis and you participated in clinical trials with a particular viral vector, you might then not be eligible to participate in subsequent clinical trials. There are certain restrictions, that's true, but I've not heard about restrictions to benefits.
Thanks, Jutta and Simon. I have a question in that you mentioned patient involvement is something that is, you know, valued within this field and hopefully going forward. In terms of kind of patient centricity in inverted commas, is that something that is being built in more to the advanced therapy environment than it, say, was into other drug development? Because now we're kind of looking backwards and trying to build patient centricity and involvement in a bit more from the start. Is that something that you would say is more of a central feature of gene therapies? And also, how do you foresee that these companies are going to build that in when you have examples like the one where, you know, there was several companies who gave up the product and actually a non-profit had to take over? How do you foresee that they're going to build that trust with those, you know, sophisticated patients or patient organizations to actually get these projects off the ground?
Yeah, I think having patient public involvement into early design of clinical trials is now an absolute must, and it certainly has increased over the last 5 years. I don't think it's necessarily different for gene therapy than it is for standard drug development, except to say that patient involvement, patients need to understand, for example, the requirements for long-term follow-up, need to understand the side effects and so on. And certainly from my experience, to have a very close link to patient groups and patient voices is absolutely essential when it comes to the design of clinical trials. What was the second part of your question related to industry?
Yeah, so just as you said, there was that treatment development that got dropped by 2 different people. I think things like that would not be conducive to building a lot of trust. And also you said that there's kind of a decreasing interest in rare diseases, which was surprising to me.
Yeah.
How can we kind of build like a strength and trust between these companies developing the gene therapies when they do take so long and they can be uncertain?
Yeah, good question. I'm not certain if I have a magic bullet here. I think maybe we're just, at the moment we have a bit of a sort of financial crisis, I think, in worldwide in biotech, and that includes gene therapy. And I really valued Simon's perspective on this as well. My take on this is that maybe pharma companies at the moment are hesitant and makes maybe some knee-jerk reactions, which over time, as the financial climate for biotech improves, might bring rare and ultra-rare diseases back on the market. But certainly within academia, you know, we retain a very strong focus on rare and ultra-rare diseases. Simon, is this something you want to add into?
Yeah, I mean, I can Just in terms of the PPIE, there's the recent grant calls, the funding calls that are coming out from LifeArc. They specify that there has to be an element of this in any application. So therefore it's mandated by funders now putting the grant calls out. In terms of the companies, so I've had a bit of experience with this over the past few years, because we found, so one of the diseases that we've worked on is a disease called dopamine transporter deficiency syndrome, which is ultra rare, but we've got very good evidence and comparison with a similar disease, that there's a very good chance that it would be effective. But it's very unattractive. It's expensive to do the trial, and it's incredibly unattractive to companies because they can't make money from it, because there aren't many patients. So even if you price it high, they're not going to make a lot of money. And so we kind of roll it together with 3 other diseases that were also rare, because the only way that we could get these to clinical trial was by attracting VC funding to be able to get the trials paid for, and it's a real it's a kind of, it's a little bit concerning that that now, you know, you're looking at the possibility that companies may actually ditch a program, but how else do we get them to the clinic? And this is certainly something that is being recognized by LifeArc, for example, where they realize that some of these rare and ultra rare diseases, they just aren't attractive to VCs and to commercial organizations, because they're not going to make a lot of money. If you're treating a disease, if you're treating an illness that everyone on the planet gets, but you're only charging, you know, 20 pence for the medicine, you're still making a lot more money than you would by charging 3 million for a patient cohort of 25 people. So we have a major problem here with trying to get these rare diseases to clinic. This is certainly something that's, you know, that some of the funders are recognizing now, and it's not a case of having a relationship with a company, because the companies are set up to make money for their shareholders. So in a way, it's kind of it's almost it's a necessary evil, but there is a bit of a problem with doing this for rare diseases, I think.
Yeah, I see that. Thank you, Jim Simon. We've only got a few minutes left, but I have one more question that I'd like to ask from the Q&A bar, which is, do you both have an opinion on the cluster effect around certain conditions where we see 2+ gene therapies being developed for specific diseases, but nothing for others?
Sorry, say that again?
Oh, sorry. Do you have an opinion on the cluster effect around certain conditions where we might be seeing 2 or more gene therapies being developed for specific diseases, but nothing for others?
Well, I mean, I've got an opinion on this, and I don't know if it's a cluster effect, but, you know, people quite often say, oh, why are there gene therapies for some diseases and not others? And my personal experience of this so far, or my perspective on this, is that for a disease to then be treated by gene therapy, but people to try to treat a disease with gene therapy, you've got to have clinicians and scientists who are sufficiently aware of the technologies to be able to get together and design a gene therapy. So we've seen this, you know, we saw this with cystic fibrosis, which has been a bit of a tough one. We've seen this with haemophilia. So haemophilia, for example, Amit Nathwan at the World Free, he understood the idea of gene therapy. He realized that haemophilia would be a great target. But something that's been astonishing for me is that rare diseases for kidney disease has hardly been touched at all, which to me has been a bit astonishing, and a little sad, because I did my PhD on kidney disease, and partly it's been because there haven't been the people there who've said, you know what, let's try doing gene therapy for these diseases. So I think what happens, you start to get a critical mass of people going, we're going to try it. And then what you need, and what the field has suffered from in the past. So in the mid 2000s, people go, you know what, we've heard this gene therapy is really good. Let's try gene therapy. But at the time, you know, full disclosure here, our technologies just weren't that good. The vectors weren't good. We weren't good at making quality or quantity. So people would try this, it would fail, and they go, oh, gene therapy is rubbish. We're gonna give it up. It doesn't work. The thing is that now, because you're getting so much better quality in terms of the sort of products that are being made, well, not products, the sort of tools that are available, and also now we have enough people in the community that believe in it, that I think this is going to start spreading out. Now, I'm not taking into consideration the fact that some diseases are actually quite hard to treat. Haemophilia is probably one of the easiest to treat, but there are a whole bunch of other diseases that are going to be tougher and are going to yet require improvements in our technology. And we're working on it at the bench, but as Uta says, it takes a long while to get these things before it will have a benefit to people.
Great, thank you, Simon. Brilliant answers, both of you, and thank you so much for those presentations as well. I know I found them really interesting and helpful, and I don't really know anything about advanced therapies, so thank you so much. So without further ado, I just want to say a huge thank you to you for coming to the webinar today, and to our brilliant speakers, Simon and Uta, and hopefully see you all again next time.